Right, great. Yeah, thank you for having us. So today we'll be talking about showing some Bayesian models and accompanying software with a brief, high-level demo of some methods we've been developing in our group over at the School of Public Health, as well as DCMB. It's on Pan-Omic data integration, integrating different types of data. That's sort of the common theme here. And so I'm Veera Baladandayuthapani. I'm a professor of biostatistics, and we'll be tag teaming with Rupam Bhattacharyya, who's now a third-year PhD student in the Department of Biostatistics and has now led one of the topics here that we'll be talking about. And Chinmay is a first-year PhD student in the Department of Biostatistics, who's going to be talking about some implementation of one of the softwares that we've been looking at for the past few months. And so just like I'll start with a very high level, almost a 70,000 high level overview of why we are doing things and also how we are doing things, to just set some context and draw some connections with the methods, more the formulation of the methods rather, that we've been sort of working on. So I mean, this is an oft repeated figure, data integration across multiple modalities that encompasses not only genomics but also other types of data. And there's a growing interest in this area, especially over the last few years. And with the key goal being how to extract biological or clinically implementable knowledge from these data sets, right? And I usually say this in some of my talks that I've given, for the last few years. Just assimilating it together, I think this might not be an understatement to say that data integration may be the key quantitative challenge that could drive modern science, and just the availability of this data, in various scales, formats, sizes, makes it really interesting as well as challenging, right? So so in part of my role, I also work with the Cancer Center. So most of my research has been focused on using cancer as a disease system. There are many bad things about cancer, but one of the good things from an analysis perspective is it's one of the most well-characterized pathophysiological or pathbiological disease systems, especially at different molecular levels. And that makes it sort of an, if not an ideal, but at least a very amenable playground to try these different kinds of integrative methods and motivate these different kinds of integrative methods. So these high throughput data now are available not only on tumor samples, but also other kinds of model systems that includes patients, cell lines, patient-derived xenografts, organoids, and that's growing day by day. And these include not only like big consortium, national and international consortiums, but also accompanying consortiums like the Cancer Imaging Archive, and as well as within our own institution with the Precision Health and the Michigan Genomics Initiative being some case examples, right? So so what at a high level, what sort of our goal in developing methods, and what sort of questions do we answer? So to contextualize that a little bit more in the context of precision oncology, I'll probably give a brief, a quick summary of what precision oncology is and where it's possibly heading. And I found this nice review paper by Schrager and Tenenbaum that sort of has this whole life cycle of precision oncology broken up by sort of three, what's precision oncology 1.0, which is probably what was 5-10 years ago, depending on small number of molecular abnormalities and always constrained by the tissue of origin or by the organ. And a 2.0 version, that's sort of where we are right now, where the number of possible actionable or clinical markers is probably in the dozens or even hundreds, and could be tissue agnostic. And 3.0 is probably you know in works on the slope for future implementation is how do we get to Pan-Omic analysis and then more quantitative statistical reverse engineering and network analysis, and that sort of completes this life cycle all the way from bench to bedside, right? So within this framework, that's where our group sits in developing methods that not only looks at different kinds of biomarker or mechanism driven discovery that's shown in this quadrant, and also look at network-based models, but also keeping in mind clinical translational applications about how do we keep that as a frame to to contextualize these models, and of course our final product in terms of software visualization and tools, right? So for today's talk, we'll be covering two methods that we've been involved in over the last few years. One is on targeted discovery, and specifically a class of models called the Ibac models, which stands for Integrative Bayesian Analysis of Genomics Model, and then briefly hit upon some generalization extensions, but that's the part that Chinmay will be showing towards the end of the talk about a Shiny app and a demo of that. And then and Rupam Bhattacharyya will be talking about a sort of a complementary as well as an orthogonal stream of work in data integration. It was how to use network models and this concept of personalized networks in cancer. What does it mean, and how do we develop those? So basically covering the whole discovery to translation cycle, and also looking at how do we calibrate drug responses, integrating data across patient cell lines and human tumors. Right. So, I'll briefly hit upon the Ibac formulation to set the context of the methods that now we'll be showing some demos for. So the Ibac conceptual framework, if we arrange, we have so the basic setting is that we have this multi-omic data at different levels that includes genomics, epigenomic, microRNA, transcriptomic, and then we have the outcomes. So if we organize it this way, and the key principle behind this is that expression, is sort of like a coalescing of these upstream molecular profiling data, and everything goes through this conduit to eventually affect the clinical outcomes. So that serves as an intermediary, and have two sets of models. One that looks at the upstream molecular profiling data and how it affects transcriptomic, which is basically the gene expression, and how that gets tied in with the clinical component of the model that ties in with the clinical outcomes and the phenotypes, and basically put the two together. And that sort of is the conceptual schema of combining these two. So in terms of the formal mathematical construction, I won't go too deep into it, but the key idea here is, so if you think about each of these data matrices being high dimensional data matrices, you have gene by patient for mRNA, you have gene by DNA level, you have the microRNAs, the methylation. And if you have clinical outcomes, which could be anything conceptually, it could be a scalar, simple like survival times, or it could be multivariate, or even image, and that's something that we worked on, actually, some of it in partnership with Arvind Rao, in extending these models to imaging phenotypes. So what we eventually, from a probabilistic perspective, what we want to actually get to is sort of like a joint distribution on all the data types. And this deconvolution of these two components into a mechanistic and a clinical model, using expression as a common, as defining that conditional representation, and having two sets of models, one driving the mechanism and other driving the clinical responses, and with some parameters that link the two models to incorporate the shared dependency. And that's the idea here. And this formulation is pretty general, so one can use any type of models. So we look at regression models because they they accord great flexibility in bringing in different types of structures. Especially if there is regression models are well utilized, in a variety of ways, and there's a lot of structure one can incorporate in terms of pathway information, a priori information in these models. But the formulation could be, for example, you could have any model here, that could, as long as this conditional representation is assumed, right? So the exact formulation of the mechanistic model, this was our original Ibac paper back in 2012. This work done by Wenting Wong and my PhD student Elizabeth Jennings. And we have different iterations of this, just improving upon the formulation of the models, making it more flexible. But the key idea here is, the first level is just a regression model that breaks the expression into multiple components driven by the upstream factors. So it's partitioning these, partitioning the cumulative expression, and it can be sort of thought of as a denoising step for the gene expression, and with the added benefit that it can identify what is sort of called driver genes. So if one of the components of the DNA is significant, then that could be a potentially DNA-driven gene or a methylation-driven gene. And in terms of parameterization, again, we keep these parameterization pretty general. So we made it as a nonlinear function. So that could be a potential nonlinear dependency between the DNA and the mRNA, or methylation and the mRNA. And the interpretation of these components is part of the mRNA that's explained by upstream platforms. Now, so what the Ibac does is basically on a gene by gene basis. So if you have a gene by number of samples, so for each gene separately, this is run. Although it could be run jointly, but that makes it easier to run it on a gene by gene, and then extract these components, the F1, F2, and F3, which are the different components, and that components actually feed into the clinical model. Before I come to that, just a caveat that this error term right here that I'm showing, so this term is not the true error as in a regression model. So it can be construed as a the estimate of the percentage mRNA that's not explained. So there could be many things that drive the expression, that we have not accounted for here, like a transcriptional factors or any other upstream platform. So anything that's not explained by these factors, gets collapsed into that epsilon term, which is.
Hey, Veera, can I ask a quick question?
Sure,
so can you add additional covariates to this, like age, or if the major subtypes of tumors, could you add that?
That's a great question. So you can, theoretically you can, but that actually comes in, that's actually a perfect segue to my next one. So those factors we added for the clinical model rather than the mechanistic model. So the belief here is that the mechanism, if we have a sample of patients, the mechanism should be pretty homogeneous. We don't have to adjust on that level. But when we want to find association with clinical phenotypes, that might become a factor. Like how well does genomics do accounting for these factors, the clinical and the demographic factors? No.
Veera, I'm surprised by your statement, because everything we've learned in the last decades is that tumors and patients with tumors are highly heterogeneous. So somehow we have to account for this marked intratumoral and intertumoral heterogeneity in all of these molecular features. In fact, as you know very well, I think the CPTAC project is about taking subcategories of organ-described tumors of origin and identifying multiple subtypes. And even then, they're highly heterogeneous.
That's right. Yeah.
How do you address that? Or do you even have a way to think about that here?
So in theory, that's easy to adjust. Like it'll be adding in the covariates at this level, and then basically having a term in here, But that's a really good question, that we don't do it with this formulation that we have here. We don't actually so this is, we can only account for inter-tumor heterogeneity because we have only, sort of one observation per tumor here.
Right.
But that's a really good, it is, it's not explicitly getting adjusted for, but implicitly, like these terms right here are individual specific. So these terms, when we do it on a gene by gene basis, so it gives me an individual specific metric or a number that can be used to translate to the clinical model. So we are implicitly doing, we're not explicitly in the mechanistic model accounting for that heterogeneity, but that's something, yeah, that's a really good point to think about, to not only incorporate in the clinical model, but in the mechanism model itself.
Right. Right.
And it could differ by stage. And when I get to the second part, I think we're doing it in a different manner, but not here. Yeah. Yeah. Thanks for the great questions. That's Maureen and Gil.
Yeah, I was just thinking that, it's very rare that you'd have all of the different omics in the mechanistic model that actually make a difference. So like if you have more information on, subtypes, that could help make up for some of the missing mechanistic data.
Yep. Yep. That's a great idea. So in the first session, we didn't think about that, but that's something moving forward, So yeah, so just carrying on, we basically the components from the mechanistic model feed into the clinical model, and that's also a standard regression model that we have. But I want to just briefly comment on the what is the interpretation of this model, because that's that sort of was the genesis of this work. And the way we think about these regression coefficients, it's not the actual DNA or the mRNA or the methylation we are bringing in. So the easiest conceptualization is thinking about genomic switches. If there is some circuitry in the cancer genome, that could be conferring a positive prognostic or a negative prognostic effect on the clinical outcomes if we think about prognosis. So there could be multiple switches that can be turned on and on. It can be the epigenomic switch, it could be the genomic switch, or it could be just possibly the transcriptomic switch. So these parameters right here corresponds to those switches. And it doesn't have to be mutually exclusive. It could be that both the genomic switch gets turned on that drives the expression and hence the survival. Or it could be the genomic switch that, that's modulated, that's the effect is modulated by. So these regression coefficients have a very specific meaning in this context of connecting it with the clinical outcomes, right? So this sits on a Bayesian model, which sort of builds on a prior set of priors that borrow strength not only across the variables but across the genes, as not only across the genes, but also a hierarchical prior formulation, and this, as I said, hit upon earlier, this accounts a lot of flexibility in terms of incorporating a lot of different types of shrinkage properties, not only pathway based but also graphical correlations, and that's some work we've been doing to incorporate that. So essentially, after all this complex model building, like what exactly goes in and comes out. So in terms of the model fitting, so this is the mechanistic version of the model, the clinical version, and what we get is a few outputs that are interpretable. One would be this integrative heat maps that tell us the percentage explained by the upstream factors. So there could be genes that are driven by these different, what percentage of upstream factors drive these gene expression. And plots like these, Manhattan plots, which tells you for each gene, not only, these are the sort of the plots of the posterior probability similar to p-value plots. What is the mechanism of action? Was this green gene driven by methylation? Was it by copy number, or was there some other mechanism? And a list of genes based on different thresholds on those posterior probabilities. Right. So as I said, there are various extensions of this that we worked on. The Ibac that will show a demo. We have a pathway-based version that takes into account different pathway level information, the external pathway level information, and a functional Ibac. Gil, you might be interested that we're working on. Maybe we can chat sometime about it. That includes RPPA and mass spec proteomics data. So it adds in one more level of proteomics to the expression, and then so it becomes a hierarchy of upstream platforms, expression, and then proteins, and then clinical outcomes. And
Yes,
then,
of course, as you know, we have the CPTAC data sets that would be very useful in applying your methods,
yeah, and
both the global proteomics and the phosphoproteomics,
right,
sometimes even other modifications.
right. And Rupam is actually that he's working on a project exactly that's characterized, and we can he'll be showing some proteomics data as part of his talk, but we can circle back on that. And then finally, shout out to Arvind, who's been a collaborator on radio. That sort of jigged our extended this to extend to radiological biomarkers as well, right? So in terms of network-based approaches, this has been a sort of a pet project of mine over the last few years on this concept of personalized networks. So when someone thinks about networks, it's usually a population level quantity. That means it's across samples. And how do we, from static data, from static molecular data, how do we get networks that could change across patients, and not only the edges, which might be the connectivity between the genes, but also their strength, and how does that change across the samples, and in this case the patients, and that trajectory might be anything, but could be disease evolution, types of cancer, or different aspects, and that's something we've been working on. And that leads to Rupam's part, where he'll show some background and work in this regard. Rupam, do you wanna?
Right. So I hope everyone can see my slides and hear me okay. Let me know if there's an issue, and I will continue from wherever Veera left it for me. So I'll just wait for a few seconds on this slide. So this is what the overall project is about. This is sort of the overarching figure that describes everything that we are doing in this. So as you can see, like from an inputs perspective, there are three things going on here. There are patient proteomic data, there are cell line proteomic data, and then also there's cell line drug sensitivity data. And it's about how we combine them, how we utilize the network information among the pathway proteomics, how we use that to do a drug sensitivity prediction, and then how do those predictions fare? How do we validate and evaluate those kind of things? So that would be sort of the overall summary of this project. So what I will be doing is that I'll show you a couple of slides on the background of this. So why we are doing this, what are the questions we're trying to answer, and what are our strategies. And then I'll show you a demo of the results that we presented in our Shiny dashboard regarding this. So let me start with the big background that we have been using. So the problem that we are trying to answer is, again, as we are described in the context of precision oncology, and we're trying to optimize treatment to each individual patient. So we are not saying that, okay, whoever has this cancer in this organ at this stage gets treated by this. We are trying to say that everyone should be characterized at a more granular level, and that granular information should inform how they are being treated using certain therapeutics or drugs or anything. So it's a relatively simpler problem from a simple data analysis perspective if you have the right kind of data. Because if you have the right kind of training data for fitting your models, which could look something like this, you have different levels of omics, you have the clinical data and all, and essentially you could make whatever model you want to make here using, say, some drug response or clinical information as your response, and these things as your covariates. You could also add in demographics or whatever you want to. And then once you have fitted that training model over a fairly representative sample, you could just use that to spit out predictive numbers for future patients that are working in. And that's what really the problem is, because it's not really that simple to have, say, for one drug, a lot of drug responses from a lot of patients, because you cannot just go ahead and treat patients with drug and see how they're responding to those things. There's usually a clinical regime involved in that and all sorts of other things. So essentially, this is the bit where we cannot have everything that we want to have in scenarios. So what do? We bring in another level of information, and that information is model systems. So things that represent a tumor as it behaves in a cellular environment, but that are outside the body that we can apply whatever we want on and then see how they respond to certain things. So of course, there have been different model systems discussed in biological literature over the years. There have been cell lines, organoids, and other things, as Veera mentioned. And of course, there are all these databases available nowadays where you can have a lot of multi-platform data for each of these things. So what our idea here is that we connect these two sources. So we connect the pharmacological data that we have for cell lines to the large-scale omics that we have for both patients and cell lines. And while doing that, we utilize also the individuality, the heterogeneity of the patients when we make the final decisions or predictions. And there are a few things that we need to take care of here that we need to account for in the model, and that's where the network part comes in. So first of all, the genes are not really acting independently, and their accumulation of effects is not really simple or linear. There are interactions and cascades going on in between them in biological functional pathways. There also are variations between and possibly within cancer lineages. So depending on what site you were looking at, you could see different behaviors. And also there are variations across model systems, because even though these cell lines are supposed to represent the patient tumors, they are in a completely different environment. They're outside the body, and they can be pretty different from how a cellular environment behaves. So we need to account for all of that inside our model somehow. So what do? So first, we just lay out what kind of data we have. So if you look at a simple 2x2 table, if I simply talk about just for proteomics for a while, this could be multi-omics as well. But if I just focus on proteomics for a while, this is roughly what you look at. And the part is this is the one that's missing. So you have these, you also have these, but then this is the one that you don't really have. So how do you fill up that gap? That's the question. And we say that we take a two-step approach. So first, we just look at the patient and the cell line proteomics. And somehow, and I'm being very vague here, we try to compare the proteomic activities, cellular similarities between these patient samples and the cell line samples. So your approach here would be saying that, suppose I give you a number of patients, which are the top matching cell lines that are similar in terms of proteomic activity with those patients. So those cell lines are what we call avatars for these patients. Now, once we have done that, then we go to the cell lines. So whatever top matching cell lines, great cell lines that we have for these patients, we build a training model for them because we have both of this data now. And once we have built that training model, now we use those to predict whatever missing red response we have here among the patients. So that's sort of how this is supposed to work in a combined two-step manner. And this part where we talk about connecting the patient proteomics and the cell line proteomics and computing sort of a similarity matrix, that's where the network comes in. Because we do it, first of all, we do it cancer by cancer. So we look at one patient cancer type, we look at all the samples, and we try to create a set of try to identify a set of cell lines that are similar to those patients. And to do that, we do our analysis pathway by pathway. So for each cancer and each pathway, you fit one global network model, and that sort of informs your population level and sample level networks. So I'm not really going into the math of that, how exactly you're doing that. I have mentioned other references down the line where you can go into it. And if you have questions about that, I can talk about that more. But roughly, these are what we get. So we get, first of all, for each sample, a pathway or network aberration score, which sort of tells us that how non-neutral, activated, or repressed a pathway is for a sample. And then using those, we compute the similarity between the patients and cell lines. And that can be done in multiple ways. Once we have those scores, you could either just compare the networks direct instead of having these scores. You could also compare the correlation between these scores, or you could do something like a unsupervised hierarchical clustering that would tell you that, okay, these cell lines are in the same cluster as these patients, so these are possibly similar, something like that. So that completes the step one. In step two, we build the response models. So we have particularly used these BART models that are known to have good selection as well as good prediction. So our response is essentially the sensitivity to a drug or a compound, and then our covariates are these pathway scores that we have already computed. So that's how we train our model and then use them to predict. And there's an existing literature and our packages and everything for that. So I've just borrowed it from there. So then I'll go into the demo, but before that, I just want to list a few resources. So this is our published paper. This is the GitHub page of our project. So we have all the codes and everything. We haven't really made it a formal package yet, but we have all the codes up there, and you can recreate all the figures in the paper. You can also run it on your own data as long as you have the format and everything correct. So give this a visit if you're interested. And then comes the demo part. So this is the shiny dashboard that I'm going to talk about for the rest of my time here. And we also have another demo video on our YouTube channel. So if you miss anything, or if you'd like to listen to any of the parts in a bit more detail, you can head over there. So I'll now change the share and just go to the dashboard and spend the rest of my time there. Right. So let me just this. So can you see my browser? Like it should be loading.
We can just see that it's processing a request.
Right, yeah. Okay. Let me just stop the sharing and do it like this. Yeah, it should be okay now. Is it?
Yes.
Okay, great. Right. So this is our shiny dashboard. This is the first page is essentially just an overview. So it talks about what things we are doing. It tells you what are the different functionalities that you have available here on this dashboard, and then what are the contents we have and all that. So let me first go to the very most granular level of results that we have here. So remember how we talked about fitting one model for each cancer and pathway combination? So we do that for both the TCGA samples and also the MCLP cell lines. And this is where you can just look at those networks. So essentially what you see in these networks is that for each pathway, the nodes are essentially the proteins that are in this network. And then the connections are determined by the posterior probabilities of those edges. So essentially, if you think of that edge sort of like a binary switch, like 1 or 0, then it essentially in the fitted Bayesian model, what is the probability of that being a 1? That's what is determining the edge strength here. And we have done this across 31 different cancers from TCGA, and also 16 different cancer tissues or sites from MCLP. So you can interact with this, you can look at the different models that we have fit, and also we have looked at 12 different functional pathways, and that was more relied on, identified based on previous literature. So you can have all those sorts of networks. You can also like click on these things and look at certain subsets of these networks and stuff. But then this is a lot of information to process, right? So what exactly do you get out of this? How exactly do you use this? So that's where the summaries come in. So we do two levels of summarizing. First of all, we do a pan-cancer summarizing sort of, where we say that for each pathway, we'll just be looking at each age. So essentially, if I just zoom in a bit, what you see here in the rows are the different ages that you could possibly have. So these are the different protein-protein pairs, and we're looking at undirected ages, so there's no ordering here. So across all the cancers, we first of all see if we can see any sort of patterns in those ages. So for example, if you see a banded line across a row, that would tell you that that's something that's present as a pan-cancer pattern. Similarly, if you see something like this as a column, then those ages are possibly turned out to be important in your model for that cancer and things like that. And also we plot the network and the network age strengths and weights and things like that. And then we go into another final level of summary. And that's when we bring in the comparison of the patient proteomics and the cell line proteomics. So the first summary is really simple. We say that two sets of cancers, one cell line, one patient, are connected via a pathway if they have, and again, I'm being a bit vague here, if they have similar level of network activity. So we have certain metrics and cutoffs for that. How you would see this figure is essentially saying that these cancers and these cancers have similar level of activity in these sets of pathways. That's sort of how we look at it. And we also did a subset of that, just looking at the tissue-specific matches. So you have a kidney cancer here and a kidney cancer here and a so on. And subsetting of that figure gives you this. You can also do a simple correlation, as I mentioned before while showing my slides. So if you do a simple correlation, this is how it would look like. So you have all the cell line cancers here, all the patient cancers here, and then the edge colors are telling you where the edges are originating from and ending in. And depending on how thick the edges are, you are knowing how the strength of that connection varies. And then comes the hierarchical clustering that I also mentioned. So this would be just simply looking at all your patients or your cell lines together, having all the pathway scores now, and just running an unsupervised clustering on them and seeing who ends up on where. So you have 3 bars here, as you can see on the rows. So this one tells you which source is the sample from. This one tells you if it's a patient sample or a cell line sample. So these darker dots are essentially the cell lines. And then this tells you the clustering. And what's interesting here that if you do this, there will be patients and cell lines who will be sharing features. So for example, if you look at cluster 4 here, there are patients and there are cell lines 4. And what we can say from this is that those patients have similar proteomic activity as these cell lines that are present. So that sort of goes into our identification about our cell lines. So this is at a global scale. This is doing it for all patients and cell lines. What you'd rather do if I had given you, say, one set of patients and told you to identify matching cell lines for them, you'd be taking those patients, you'd be taking all the cell lines, run a clustering like this and see which cell lines end up together with those patients. And then that would be sort of your matching set that goes into the next step. So now comes the final step. Now comes the...
May I ask you about these heat maps?
Yeah, sure.
I'm trying to see if which of these tumor types by anatomy
So
look
types are
more homogeneous than others. So maybe C8 is a good example.
Right.
What is C8? I don't know what that is.
C8 is this, and C8 is breast cancer. So this is right here.
But breast cancer, that's very surprising because we know there are at least 3 major different categories of breast cancers.
Right. So you can see here, like C8 is this color, and the same color appears here in a different cluster as well. So roughly speaking, and then there are a few of them that are hanging around here at C18. So this is sort of like the larger subset of the BRCA samples that we have. This is another, and then we have a few here.
So the breast is dark blue. Is that what that color is?
Right. Yeah, if you would call that so. Yeah, that's essentially this right here.
So these numbered clusters are not by anatomy. These are by pathways.
These are just by the pathway scores that we have identified.
Yeah.
So when we are running the clustering, no information about where the samples come from goes into there. It's completely unsupervised.
Mm-hmm.
Yeah. And Gil, to your point, actually, we also did an analysis, not in this paper, but a previous iteration of this, where we looked at the classifications of the breast cancer by the different subtypes. I think it's the Peru subtypes, the ER positive, and looking at pathway scores and how well they are prognostic. No, if we had known subtypes, that was a more supervised analysis. No, and we did find some differences. Between the different subtypes using the pathway scores.
Right. So on the vertical here, some of these pathways are big processes like apoptosis and cell cycle and so forth. The one that jumps off the page, I guess, is RAS, which is in a great many different tumors, but still a little bit heterogeneous. There are only a few like that, RAS, RTK. Of course, RTK has a lot of different kinds of receptor kinases, isn't it?
Right. So these were the pathways that mapped from the RPPA data. So under the hood, this is using that antibody panel.
That's a real problem, I think. I've never done too much with that data set because I didn't trust the method, and it doesn't capture quantitation of very many proteins.
Yep.
So the CPTAC is the new generation of data, I would say.
Easily. I mean, those are sort of like, for lack of a better word, cherry-picked proteins, and which are clinically actionable. So it's only around 200 to 400 antibodies. I think they've increased it to 400 now. So the set we worked with is, I think, is around 200, 300, and that's the limited set of proteins.
Right.
I think the version that I used for RPPA was like 200 odd proteins. And then when you subset it, map it to these pathways, I think we had like 75 or 80, something like that. And also, like, going back to the point of like subtyping and stuff, we also did one small supplementary analysis here, which I didn't mention. But so for the head and neck samples, these HNSC samples, we saw that they were sort of like broken up into two different groups. So like one here and then one here. What we did was we just did, so this is unsupervised clustering, and that breakup is completely unsupervised as well. What we simply did is just we compared those two subgroups of patients in terms of their survival, which is also available from TCGA. And we saw that there was a clear separation by doing a simple comparison or simple testing.
So how else are those two subgroups different?
So these two subgroups that we saw here, so for example, if you look at say HNSC here, this color, then these are the subsets that are identified by the clustering. So the clustering doesn't know about where these patients come from, but it only knows about these pathway activities. What we compared between them was the survival. We didn't go as much into like their other information that might be available, like clinically how different they were and things like that. But yeah, possibly that could be one other investigation that could be.
Well, the two big variables for head and neck, I think, one is HPV, the human papillomavirus positive versus HPV negative. And we've done them separately in the CP-TAC. Very different. The other is anatomical, because head and neck covers a lot of different structures, from the tongue to the larynx and the pharynx and so forth. And there are different subsets depending on which combinations of anatomy. And that information is often available. That's part of your clinical parameters, I hope.
Yeah. I think we did have the HPV. I don't think we had the anatomy as much, right, Rupam?
We did the HPV. It was also present in the previous paper that Veera mentioned as well. I think there also we did something similar, just as a survival. And again, there the difference was pretty significant, but I don't think we did the anatomical one, right?
Well, our CP-TAC paper on HPV negative head and neck squamous is out in Cancer Cell. If you want to look for it, it's a good paper.
Sure. Okay, so let me go into the last bit of this demo. So now we come to how we train this model. So we've sort of looked at this into two different subsets of information. So of course, like for a given set of cancer patients, you can identify different sets of matching cell lines, and then you can train models on those. So we sort of try to train models on the different tissues that we had for the cell lines and try to see what pathways were turning out to be higher important predictors in the BART models that we were fitting. So the first few plots that we'll see here, including the spider plot, and then these pathway networks, they are mostly summarizing those things. So you are essentially trying to sort of rank that in which tissue, which pathway is becoming a more important predictor. And again, we are doing it across all layers of information. So you can look at the different cell lines that we have. You can also look at the different drugs that we have been using and things like that. And then accordingly, you can go into the final drug response prediction. Now here comes the tricky part, that after you predict drug responses for patients, this was a question that we spent a bit of time with, that how do you validate those? How do you really know that you are actually doing something sensible? So we sort of did two things. One that's presented here is that we ranked these patient cancers corresponding to drugs, depending on how much predicted sensitivity we had for each drug. So these percentages that you see is the percentage of patients who have greater than 0.5 predicted sensitivity, because we are talking about a 0-1 scale here. And what we found that the top ranking drugs corresponding to the different patient cancers are actually some that are quite well known in the literature, and they are actually used in a true clinical setting to treat those cancers. So that was sort of like one literature-based validation that we did. We also did some other validation with some drugs sensitivity data that we had from TCGA, and they're also the typical metrics that we use, like AUCs and stuff, turned out to be really good. So
are these percentages for cell lines or for patients?
These are for patients. So for a patient cancer, suppose you have n number of samples, what proportion of that n number of samples are predicted to have higher sensitivity?
This is quite astonishing. There are very few drugs for anything. They work in 100% of patients or 90% of patients.
Yeah, so that really depends on the cutoff here. So because this was on a 0-1 scale that we were predicting, we decided the cutoff of having a greater than 0.5 sensitivity. So depending on how you are tuning this from your final model, these numbers may be changing. But I guess what's more important than these absolute numbers here is that is this relative sort of ranking of drugs. That's sort of the message that we try to give from here. And
I don't know.
so, and then there's this final page where you can look into our GitHub page. You can also download all the codes and everything. So that's where I will finish with my part and hand it over to Chinmay.
And while Chinmay is loading up, Gil, to your point, so this was sort of, you have to take this with a pinch of salt. The reason being that this is from the TCGA data. So the TCGA clinical files have what drugs the patient actually got and as part of that treatment. So this was like I would say a mini validation. So do we at least get that right? No.
Well, when you say they got it, does it mean that they responded well to it or just received the prescription?
We don't know. They just got it. We don't know if they responded or not. All we know is they got it.
But this word sensitivity implies more than that.
Right. So, yeah, I don't think that's the interpretation here. It's just like, like a backwards validation saying, okay, they got it, did we get that right? No. Yeah. I mean, a true validation would be on a clinical trial where we know that this drug was given and we know the sensitivity, And
well, for all those drugs, nearly all those drugs have been through a lot of clinical trials, actually.
And corresponding genomics data at baseline,
Right.
like the proteomics or maybe run this through with an expression.
Yeah, proteomics less so, right.
All right, well, thank you, Rupam, for that presentation. And I'll go on to talk about kind of what Veera was touching upon earlier regarding the Ibac framework and what we've done to sort of make it more accessible for people. So I've been working on a Shiny application to sort of introduce the Ibac framework and allow people to fit their own models and identify driver genes. So this is our Shiny application, and you can access it and then actually input your own data. So we have a data input page where we take in CSV files for copy number variant data, methylation data, and mRNA expression data. Now this is used to build the mechanistic model, and then you can also input clinical response. So I've already inputted a bunch of data in just to speed up the process. And the data I've inputted in is a glioblastoma data set. So this will be genes associated with glioblastoma and then survival times of cases of glioblastoma. And after running the analysis, what the Ibac framework will produce for you is a bunch of posterior probability plots for the coefficients of the various different genes along with their corresponding genomic data source, so methylation, copy number variant. And this will tell you the probability that the gene, that the data source associated with a given gene results in a certain percentage of increased survival time for the top plot and decreased survival time for the bottom plot. And this slider you can actually change. So if you're looking for any genes with a 20% increase, obviously that's going to be very rare, but you can adjust the slider as you will to identify these genes, at least through the posterior probability plots. Then you can visit the gene tables, and this will actually just give you the list of genes which pass through the thresholding that you selected through the posterior probability plots. So in this case, gene PIK3R2 is known to be significant, at least regarding its methylation data, for positive effect on the outcome. And then it'll also find the genes which are associated with negative effects and their corresponding data source. And then the final plot that we produce for you is a heat map, which tells you the proportion of variability explained by the various different data sources in the mechanistic model. So you'll notice that most of the genes, the methylation data doesn't really give too much information or give too much signal, but for a couple of genes at the bottom, such as EGFR, the copy number is a lot of information, about 75% into the values for the mRNA data. This final column just lets you know, like, okay, there's additional information that is not able to be captured by the mechanistic model, but it still exists. And this will help you identify which data source contributes to the effect of the certain genes.
So on this slide, why is copy number and mRNA, why are they in opposite directions?
So in the mechanistic model, you have the methylation, copy number, and other genomic sources explaining mRNA. So the reason why they're in opposite directions is because if a lot of variation is explained by copy number variant, there's not much explained by the other sources.
So that's the leftover part, Gil. I was talking in the construction of this model. This what's left over. Is it pure, like whatever is left over? Is it purely transcriptional, or is it something else? No. If the upstream platforms, the methylation and copy number don't explain it, then we can attribute it just to the gene expression. So it's an artifact of the construction of the model, because if copy number explains it, takes it away from what's remaining. No.
Okay. I don't know if that's good or bad. Just don't know.
It's one interpretation. It's just from an interpretational perspective. That so if you think about like the whole gene's methylation is say 100%. So what this plot shows is is there any how much of that percentage is explained by methylation? What percentage is attributable to copy number, and what's left over? And then you can add in other variables too, so you can expand on the columns to get to other type of other upstream platform data as well. For example, mutation or transcription factor, those kind of things as well. So it's just basically a sort of a deconvolution plot of that.
Yeah. Okay.
That's the interpretation here. No.
I was wondering if you could say a little bit about the recommended minimum sample size for Ibac in order to get useful results from it. Because in my experience with these multi-omics integration methods, they tend to require pretty large sample sizes, and unfortunately, it's usually larger than what we have. I suppose it probably depends on whether your outcome is survival or binary.
Correct. That's exactly right, Maureen. So the good thing is for the mechanistic model, I think it's okay to even go with a lower sample size. So essentially we are fitting a model with 3 covariates, right? It's on a per gene basis. So there actually we can have a lot of, a reasonable sample size will work really well, because it's running on a gene by gene basis. I think that the sample size requirements factor in much more importantly if it's in the for the clinical model, because that's run across genes, because trying to find a signature, and I totally agree. I think you need at least a good enough sample size, to have. It's the same sort of argument for any regression model when you're doing variable selection or feature selection, to have enough power to detect these. And especially for survival, it's even harder, because then you have to because it's an effect on the prognosis, and what percentage change do you want to see in terms
right
of.
So are you thinking like you would need hundreds for
So the data sets we have test driven is on the TCGA scale, so usually hundreds to a few hundreds, that would be a good test case. Anything below, yeah, you'll get something, but how much confidence do you put in that?
Yeah.
So the good thing here is, what we also didn't show is there's a multiplicity correction also going on with these posterior probabilities that we also do. So that's another added level of, at least statistical protection, in terms of controlling multiplicities that one can do, especially for smaller sample sizes.
I guess I'll stop and kind of conclude the presentation. So these are all the links to our resources. So if you want to view the transfer size Shiny app, it's all on the bayesrx.shinyapps.io server. And yeah, thank you for your attention. Any general questions or comments?
Joint presentation.
Thanks, Gil.
Thank you.